All right, so this last part, Roman numeral 4, mutations of chapter 10. Mutations are fun to talk about because I think people have ideas of what mutations are, and these like two-headed monsters or three-eyed toads. But really what mutations are, in the very basic definition of it, is a change in the base sequence of DNA. If you think about it, how can you change the organism if you don't change the DNA, right? The DNA is what codes for the information that makes who you are. So to fundamentally change something, whether it's a plant or an animal or a bacteria, you have to change the DNA sequence, right? You have to edit what you're coding for. And so that is, when it happens in nature, it happens randomly. You can get these sequences of DNA changed, and we'll talk about the different mechanisms, but these mutations are completely random. They are mistakes, and the fallout of those mistakes can be, good or bad, and it's basically the origin of evolution, because how can you change the organism without changing the DNA? All right, so let's take a look at the types of mutations we have. So letter A and letter B are the two big classes of mutations. So letter A says base pair substitution mutations or point mutations, which is this box here. What's happening in this box is like, what happens if you just change one base? One letter is altered, okay? So you're just substituting out one base for another base. So for example, a G is replaced with a T. That's called a point mutation as well, because that's just one letter or one point. And it turns out if you just change one letter, you can have different scenarios emerge, which is a silent mutation, and a sense mutation, or a nonsense mutation. So let's just look at this first, okay? Let's look at our picture that we have in your notes. So this is kind of like our normal DNA double strand. The normal route of replication, if this was completely normal, transcribed, translated, we have a normal protein. Normal is called wild type. So this is going to be our most prominent functional protein, right? Wild type. All right, well, what happens if there's a mutation? Let's look at first our silent, all right? So there is a mutation. The TAC is now a TAT, right? When you transcribe that, you get a different, you get the same amino acid coded for, and you get the same protein. This is a silent mutation. This can happen. You actually change a letter of DNA, but the fallout is you get the same amino acid, the same codon. It's not the same codon. It's UAU, UAC. They're not the same codon, but you do get the same amino acid. So let's look at why, right? Remember when there is this redundancy of the genetic code. So let's look at tyrosine. It turns out there's two codons that both code for tyrosine. So you can swap out that letter. It could be a U or it could be a C, and you could still code for tyrosine, and nothing changes about that protein. Proline, if you look at proline, as long as you have two C's in a row, the last letter can be anything, and you still code for proline. Okay? So the fact that this letter changed and you still have the same amino acid did not change the protein at all, and so that's called a silent mutation. The mutation happened, but it's completely silent. You don't know. Let's look at the other category here. Nonsense. Let's start with nonsense since it's the next in a row. We have a mutation occurring, and instead of tyrosine, you get a stop codon. That's strange, right? So remember, stop codons. Let's go back. Oops. Sorry about that. There are 3 stop codons encoded for in the genetic code. Those stop codons, if you remember what they're used for, they're to stop translation. So as the ribosome reads RNA, it stops at a stop codon. Those don't code for anything. They're stop messages, right? And remember, we also had a start, but the start also codes for methionine. Well, let's look, so if we are converting what we should be coding for tyrosine, but we actually accidentally coded for a stop, what's going to happen to your protein? It stops getting made. You get an incomplete protein, all right? So this is called a nonsense mutation when you code for a stop. Lastly, we have something called a missense mutation. You get a change in the letter, right, one base, and the normal wild type, which was tyrosine, now changed to asparagine. So you're coding for a completely different amino acid. What you get is a protein. You get a fully made protein, but there is a substitute for an amino acid. Now, this is missense. That mistake can be good or bad or meh, okay? Because it just depends. The example I have in your notes is sickle cell anemia is actually this kind of a mutation. Sickle cell anemia is a point mutation or a base substitution mutation where one amino acid gets swapped out, and that one amino acid played a huge role in the functionality of hemoglobin protein. But other times you get a change in amino acid and it's not as drastic. Or sometimes the protein might change function. You don't know. So these missense mutations are a little bit of a question mark as to what they're going to do for the protein. So what I have in the notes is the outcomes for effects on the protein can vary. However, I do have sickle cell anemia as an example of a very bad outcome. All right, so this, the base substitution, right, we just looked at this. So here's our normal gene. We can replace one letter. Here's glycine. It changed to serine. How would you classify this? Is this a silent mutation? Is it a nonsense mutation? Or do you think it's a missense mutation? So if you said missense, because we changed the amino acid, you are correct. That's a missense mutation. What we're going to turn our attention to is now letter B, frame shift mutations. So I'm going to go back up to this picture. So insertions or deletions of DNA nucleotides. This box is also known as frame shifts. Okay, so let's take the sentence, the cat sat. That's a sentence made with 3-letter words, right? The 3-letter words being codons. If I took out the H, right, of the cat sat, we would not have the same letter or the same sentence, and the reading frame, so you know how codons are read in groups of 3, then the reading frame is off because instead of THECAT, you're going to read TEC. That's your first codon. The second codon is also messed up, and the third codon is also messed up. So you can see that if I deleted a base, that it's not just that one codon that's going to be affected, it's all the codons downstream from that. Okay, so in this picture here, we deleted a letter. Okay, so we're looking at, this is kind of our base here. We have methionine, we have lysine, but instead of just changing serine and the alanine remaining the same, we made leucine, which is a mistake, and then actually alanine is coded for again, but that's just kind of a weird example, but they're both affected. And then if you randomly put in a nucleotide, then it's going to have the same effect. You're going to shift the reading frame. So you're going to have leucine, then tryptophan, then arginine. Every single codon from the point on is going to be altered. Okay, so in terms of like what's worse for an organism to have, it's probably frame shift, right? Because inserting or deleting, you're not only just changing that one codon, you're changing all the codons from that point down. So this is our example of sickle cell. I was talking about this, but I didn't show you the picture. So this is our wild type. Here we have our mutated point here. So this is a base substitution. There's only one letter that's changed. The mRNA changes, and then our protein, our amino acid, also changes. So again, this change is a missense, and it causes that protein to really fold abnormally. So this point of the amino acid is a very critical point for the whole protein. All right, so are mutations bad? Yeah, most of the time, yes. I would say most of the time they are sort of bad for you to accumulate mutations, but they are also helpful, and they are the basis of evolution. And I think this is our sort of picture of what mutations can cause. The first bolded sentence, right, I have in the notes is mutations are random, which is listed here. Mutations are random. An organism like you or I, or a bug, or ants, or bacteria, or whatever it is, plants out there, they can't decide to mutate a certain gene. That's just not possible. So when DNA is mutated, when there's a mistake that occurs in building DNA, then it's random on where it occurs on that DNA. So in order for mutations to be helpful, it's a completely random event. So let me give you an example of here. So these are some lice. These lice have been becoming more and more resistant to the shampoos that people use to kill lice on their hair. And randomly, just randomly, there is one louse, I guess, that's resistant to the shampoo, because when the DNA was being copied, when this louse was very small and just an embryo, there was a mistake that occurred, and the mistake just happened to be in a protein that caused, this louse to be sort of susceptible to the shampoo. This resistant form will be better fit to live in your hair, right? Because if you use the shampoo, you are going to kill off the non-resistant lice, right? They're dead because the shampoo works on them. But because of the random mutation that occurred in this one's going to live. And because it's alive, it's going to reproduce. And the genes, that mutated gene that it got randomly, will be passed on to its offspring. And those offspring will survive and pass it on to their offspring, and so on and so forth. So the initial mutation that occurred was random, but since it was beneficial for these lice, it survives, right? So it gets passed on to their offspring, and then all of a sudden you have a new population of lice with resistance to the shampoo. So are mutations bad? For this lice example, no. Mutations are very, very helpful, and they help the lice survive. And this is exactly the same way that you get antibiotic-resistant strains of bacteria. There is some bacteria randomly, just by accident, that have a mutation in their DNA that allows them to survive antibiotics. And because they are surviving the antibiotics, they survive, they reproduce, they pass it on to their offspring, and every single offspring from there on out will be resistant to the antibiotic. And that's just how it happens. So are resistance bad? Most of the time, yes. And some of the time, it's actually very, very beneficial for an organism. What causes mutations? So unfortunately, mutations can be spontaneous, number one. This is when you copy DNA. The DNA polymerase, which is very good at proofreading, is not perfect at proofreading, and there's a mistake that happens. So spontaneous mutations are mistakes that happen randomly when you copy DNA. Number two, mutagens. Okay, which is listed as number one in this picture. Mutagens are actual either radiation or some kind of chemical that causes the DNA to have mistakes in it, that directly or indirectly cause mutations. So in the notes, I have listed for you some chemicals, and I have listed for you some chemicals here. So like alcoholic beverages, arsenic, asbestos, but also, acetaldehyde, aflatoxins, this is from mold, but also there are viruses, right? Epstein-Barr, human papillomavirus, hepatitis C. Those can cause, when you're infected with those viruses, they can actually push for changes in the DNA and cause mistakes to build up in your DNA. Radiation, we know that UV rays can be very harmful to your body. They can cause skin cancer, which is basically a buildup of mutations. So tobacco, we know tobacco can cause lung cancer. Cancers, by the way, are all, it's a disease of mutations. And we'll talk about more about this when we do cell reproduction. But when your cells reproduce, they do it in a very controlled way, dictated by healthy genes. And if you mutated those genes that controlled the cell's division process, then the cells can divide uncontrollably, and you get big clumps of cells in your body, and that's basically what a tumor is. And then those tumors can travel through the body, and that's a metastatic tumor, and that is cancer. So chemicals, so why do I have this list here? Chemicals that have been proven without a reasonable doubt, right, to cause mutations are given the name carcinogens. So these are definite bad things for you, carcinogens. Radiation, you can't do anything to stay away from the sun, right? You can wear sunscreen, but it is definitely something that can cause mutations in your DNA. So in short, in your outline, 2 classifications of mutagens, chemicals and radiation. Mutagens, these chemicals that are known 100% to cause cancer, is called a carcinogen. Okay, so the last part of this before we answer this question is mutations and evolution. Even though mutations can be harmful, they can also be extremely helpful in both nature and in the lab. Mutations are also very useful tools for geneticists to track evolution and to evaluate whether a substance is mutagenic or not. And mutations make evolution occur by natural selection. Mutations are the basis for life's diversity, right? Again, you can't change the organism unless you change the DNA. All right, so let's answer this question. A geneticist finds that a particular mutation has no effect on the polypeptide encoded by the gene. This mutation is probably what? Or probably involves what? So if you said letter D, this is correct. If it has no effect on the protein or on the polypeptide, that means that none of the amino acids changed. If you delete or insert a nucleotide, you're going to affect the codon, and you're going to affect every codon from that point on. A and B are frame shift mutations, so that cannot be an answer. If you change the start codon, remember the start codon dictates where you start making the protein in translation. That will drastically alter your protein, right? Because how do you know where to start? So the only thing that would have no effect is a substitution of a nucleotide. This is a base pair substitution. And this would be under the silent mutation, right? You can change a base, but it doesn't necessarily mean that you change the codon. I'm sorry, it doesn't necessarily mean that you change the amino acid coded for. All right, which is mismatched? Okay, the answer is A, because what DNA polymerase does is replicate. DNA polymerase makes DNA, RNA polymerase makes RNA. So DNA polymerase, the matching part here should be replication, DNA replication. What the enzyme is called that it does transcription is RNA polymerase, because RNA polymerase makes RNA, and transcription is the process that makes RNA. All right, and that's it.